Good afternoon, good evening, everyone. I'm Ruturaj Govekar, and I welcome you all on behalf of CCAMP. CCAMP is India's premier life science innovation and entrepreneurship and technology hub, as well as one of the largest life science incubators in the country. CCAMP provides funding, incubation, acceleration, and mentorship to startups and has built a vibrant ecosystem to nurture deep science-driven enterprises. It's an initiative of the Ministry of Science and Technology, Government of India, and is located in Bangalore. And CCAMP has funded and incubated and mentored over 500 startups in recent years. Today's conversation on use of sequencing technologies to address antimicrobial resistance is part of the series of webinars we organize every month as a CARBEX GAN activity. CARBEX stands for Combating Antibiotic Resistance Bacteria Biopharmaceutical Accelerator and is a global non-profit partnership focused on supporting antibacterial research. CARBEX is a global network of accelerators working in various fields of AMR, and CCAMP is one of the only member of this global network of accelerators outside of US and Europe. Our idea of organizing these webinars or conversations every month is to stimulate conversations around innovations in technology suitable to combat AMR. We support deep tech startups, and our effort always is to include expert inputs from academia, industry, and entrepreneurs. In line with this, we have two such speakers, one coming from both obviously deeply rooted in academia, but one more so in academia, and the other who has taken a step in the direction of entrepreneurship and has successfully founded a commercial venture using deep tech at its heart. Let me introduce you to the first speaker, and that is Professor Yogesh Shouche. He was a research scientist at National Center for Cell Sciences, that is NCCS Pune, an autonomous institute under the DBT, Government of India. He was there for over 30 years. His major area of interest is microbial communities in unique ecosystems and how they interact with each other and their surroundings. In the last few years, his group has worked extensively on human microbiome of the Indian population and how it is affected by lifestyles and genetics. They have also tried to understand microbiome alteration in diseases like diabetes, obesity, celiac disease, with the aim of developing microbiome-based therapeutic solutions. At NCCS, he also developed a microbial culture collection that harbors microbes from different ecosystems and also has certain specialized microbes like anoxygenic photosynthetic bacteria, antibiotic-resistant bacteria, and anaerobes. This is the largest culture collection globally under one roof and is also an international depository authority, that is an IDA. It's an IDA facility under the Budapest Treaty and under the Biological Diversity Act of India as well. The other microbial ecosystems that he has worked on are effluent treatment plants, impact of mass bathing events such as Kumbh Mela on river ecosystems, and stratosphere, besides microbial resistance to antimicrobials and antibiotics. He has keen interest in science popularization and has written several articles in English as well as Marathi newspapers and magazines, besides giving talks on radio at schools, colleges, and at public events. I welcome you, Dr. Shouche. Our second speaker for today is Dr. Anirvan Chatterjee. He's a data scientist and a researcher who has spent more than 15 years in infectious disease and genomics. As a researcher, he has been involved in several high-impact projects, including his work at Oxford University, where he was part of the team that created and deployed practically the world's first clinical genomic solution for a disease at a population level. He also pioneered the use of whole genome sequencing for tuberculosis diagnostics and treatment. Additionally, Dr. Anirvan was instrumental in including antimicrobial resistance, that is AMR, as one of the Sustainable Development Goals, SDGs, under the UN Charter through a global water study. Returning to India, he introduced whole genome sequencing technology for tuberculosis elimination and co-founded Haystacks Analytics with his childhood friend Gaurav Srivastava and Professor Kiran, his mentor from IIT Bombay, who specializes in evolutionary biology and genomics. I welcome both the speakers and also our audience. So we are going to keep this completely interactive. But before the audience sort of warms up, we will have some icebreaker questions from in-house. So I will start with my first question. But before that, I would like Professor Yogesh Shouche as well as Anirvan, hi, to both maybe set the context and start with general thoughts in this field and your experience working in this field collectively over, I guess, 5 decades. Thank you.
Yeah. So can I begin or?
Yes, please.
Yeah. Okay. So since our aim to have the session more interactive, I'll be very brief in what I'm going to say. And since the theme is today NGS and AMR, I would just like to say that ever since the NGS technologies have developed, we have come a long way. And today we all know that AMR is a major global health concern, more so for India for various reasons. And NGS technology is or already playing and going to play a major role in all aspects of AMR. So it includes accurate diagnosis of AMR profile as well as the causative agent, understanding the mechanism behind AMR, understanding the spread of AMR and evolution of AMR. So all these aspects, NGS technologies are going to play a major role. And some of the aspects of this we would be discussing during this session. So as and when we come across a question or some query, I will give you some examples on all aspects of this during the discussion. So I won't say anything further. I'll leave it more for the discussion. Thank you.
Thank you. Dr. Anirvan.
Yeah, hi. Again, first of all, thanks, Ruturaj and the CCAM team for inviting me, and can't explain what an honor it is to have this discussion with Professor Yogesh Shouche. I think working in genomics, he has been almost a godfather figure in metagenomics, and so it's an amazing honor, sir. Instead of trying to tell anything, let me just get to the point as to a small story about how I ended up starting Haystack. Because Professor Kiran and I were working on isolating viruses, giant viruses from sewage. And these viruses, actually, some of these viruses grow within amoeba. So we had an amoeba culture. So we take the sewage and put it into amoeba culture. Now you put anything in culture, everything else will also grow. So our job was to put in antibiotics in that media and suppress the growth of bacteria and fungi. Remember, my focus was to discover viruses. What I ended up seeing was we threw some 25 different antibiotics on that culture, and there would still be colonies. And I think that is the size of the problem that we're talking about is that big. And we are not talking about a niche environment, nothing. This is Powai Lake and sewage from outside of IIT Bombay, right here. So I think that's where things are. Very frankly speaking, it has long been called as a silent epidemic. I don't think it is silent. And I much congratulate the Carbex initiative and CCAMP and others who have been supported on this, because I do believe that even if we want to get anywhere close to getting to some solutions on antibiotic resistance, it will require a humongous amount of collaborative effort for people of different stakeholders to come together with the single intent that we need to save the antibiotics. So that's the entire problem with antimicrobial resistance. If I could explain that to you in a short story, good for all of us. Otherwise, up to you, Ruturaj. Now we have all the other questions that we'll be discussing.
Thanks. I mean, I guess the context has been set very wonderfully. Professor Shouche, I would just like to have a general question which can serve as an icebreaker, and then I hope this will stimulate the audience to start raising their hands to ask the question, or they can also type if they feel that is more suitable. So just for that, it's a question, a very general question in the sense, like, because your experience comes from bacterial genomics and then of course AMR, what are the advantages of these sequencing technologies that you see, especially in the field of bacterial genomics and AMR, And do you have any particular examples from your vast experience where it was some discovery or some insight was only possible because we had access to sequencing technologies?
That's a very interesting question, Ruturaj. And Anirvan said I'm a godfather or whatever. I don't know whether I'm godfather or not, but I'm definitely an old man in science. And talking about sequencing technology in my career, I have seen all the transitions in the sequencing. So when I was a PhD student, or even in the beginning of my career, the sequencing was conventional radioactivity and autoradiography based sequencing. So I started from there and reached today where we have single molecule DNA sequencing technologies. So whatever transitions have happened so far, I have seen all of those transitions. And there's a reason I can comment that how remarkably the technologies have changed over the last 20 to 30 years, and how they have become faster, quicker, more accurate, and high throughput. So all the the developments which you see today would not have been possible without NGS technologies. Because earlier, when I started my career as a PhD student, you sequence one gene somehow and you would get a Nature paper. That was the situation those days. So from there, we have moved on to genome sequencing, where again, initial days, it was a very laborious and expensive exercise. I remember when first microbial genome, that is bacterial Haemophilus influenzae, was sequenced, it took almost 1 year and 100 machines available that time to do that sequence. So from there, today we have come to a situation where a single machine can give us several hundred human genome sequences in a matter of 24 to 48 hours. So that's how these technologies have changed dramatically. They have changed dramatically in terms of output, the time taken, and of course the cost. And typically, when human genome sequencing project was proposed at the beginning of this century, it was to take 11 years and something like 15,000 crore rupees was the cost for a single human genome. Now, as I said, we can do several hundred human genomes in a matter of few hours, and the cost of as less as 50 to 60 thousand rupees, or at the most 1 lakh rupees, we can do that. So that has been a dramatic change. And then in terms of handling the samples, in terms of accuracy and reproducibility, it has been a long leap. I'll not go into the technological details of the techniques, because that is not probably relevant here today. But the point I would emphasize is that they have improved dramatically in terms of time taken, the amount of output, and the cost. And here I would say that prima facie, the cost, it may appear to be very expensive, but if you look at the per sample cost, it is much less than what we had started with. So they are becoming more and more affordable. Today, as you know, we have reached a stage where we have bedside sequencing technology available. The sample really need not have to go to some sophisticated lab or even a separate lab in the hospital. Technically, we can do a bedside diagnostic using a smaller pen drive size DNA sequencer. So we have reached that stage today in 30 years. So that's the remarkable development which we have seen in last 2 to 3 decades. And coming to examples, there are many examples that can be given about how these technologies have helped. There are today in many places they use, for example, the one example which I know of is neonatal sepsis. So the preterm babies are more likely to develop neonatal sepsis. So what people are doing now is that for the preterm babies, right from their birth, they monitor the using clinical metagenomics, they monitor the microbiome as well as AMR genes in that, so that they can make diagnosis of likelihood of development of sepsis even before the infection occurs. So those studies are already on. And if you look at the literature, there are umpteen number of examples. Like recently, there was a paper published wherein there was an outbreak of MRSA in neonatal unit, neonatal intensive care unit, and the source of infection was traced to a hospital worker using NGS technologies. So that's how these technologies are very powerful and extremely useful in determining the resistance pattern as well as tracing the source of the infection.
Thank you. I will ask another ice-breaking question to Anirvan, and then I see the audience is warming up, and then I will open it to the audience. So, Dr. Anirvan, you have a sort of commercial products for TB as well as sepsis. So, I wanted to ask you, what are the similarities and differences in these approaching, devising diagnostic tools for these two diseases? Can you sort of slightly change here and focus on these two diseases?
Sure. Interesting thing. Okay, let's start from here. The tuberculosis as a bacteria is a very interesting organism. It is not known to have extra chromosomal inheritance. And whereas when we talk of bacterial genomes, a lot of the antimicrobial resistance is encoded in extra chromosomal inheritance. So while they're both bacteria, the approaches are wildly different. But then when we developed this, this was deeply connected. And it was, again, a very chance event. So when we started doing tuberculosis genome sequencing, whole genome sequencing of patients from across India, we found that about 2.5 to 3.5% of classified TB patients undergoing anti-TB therapy did not even have tuberculosis. They had non-tuberculosis mycobacterial infection and several other kinds of infection. Now, as Professor Yogesh was saying, we were doing whole genome. The advantage of doing whole genome is, and we have always stuck to doing whole genome at Haystack because we wanted to bring, okay, our main hypothesis is, we asked the question, if something is the best standard in research, why should it not be the standard of care in clinical practice? That's the simple question we are asking. And that's the same thing with sepsis. And I'll come to that in a bit. So because we're doing whole genome sequencing, we started detecting non-tuberculosis mycobacteria. And then we realized that this can actually be opened up. The TB test can be opened up. But the problem statement was very, very different. A TB therapy lasts for more than 6 to 8 months, sometimes more than a year. It requires multi-drug therapy. The patient is not always super critical, at least towards the beginning and even mid-time of the disease. But when we talk of sepsis, the patient is critical. Like in sepsis, the patient is already critical. And now, here are the technological differences. When you're looking for drug resistance in tuberculosis, you're primarily looking for SNPs and indels. Now, if you want to look at SNPs and indels, you want to do short-read sequencing, you want to do shotgun sequencing, you want to do very high-depth sequencing, which means you're moving into instruments like Illumina and BGI, which are extremely expensive to set up and also very expensive to run. Whereas when you're looking for drug resistance in bacteria and otherwise, other bacteria, you're looking for cassettes. You're not primarily looking for SNPs. There are a whole class of SNPs as well, but you're looking for cassettes. And with these things in mind, what we realized is the TB genomic solution we were able to scale by solving a problem. How do we get a large number of samples into a single sequencing run? That's the problem statement. There is nothing else in TB. The problem statement was, how can we get a large number of sequences in a single sequencing run, week on week, day on day? So what we did is, we went ahead and partnered with literally the entire TB culture ecosystem. We partnered with the intermediate reference laboratories, the national reference laboratories, with ThyroCare, Metropolis, SRL, Lalpath, Sterling Accuracy, AIMS, every hospital. We went ahead and partnered with them and said, you know what, don't change anything. You bring your samples, run your sputum, enrich them, extract the DNA, send it to the sequencer. In less than a year, we had already done some 5,000 TB genomes of clinical samples, which I hadn't been able to do in 7 years of projects that I had written with DBT. That is the beauty of scale. Now, in this system, as you realize, the logistics is sample gets collected, comes to a lab, then comes to a sequencer, then the results go back. This takes time. In sepsis, that's the one thing we don't have. So then we moved this entire bioinformatics into long read sequencing. And when we started moving into long read sequencing, we found out that now we can implant a sequencer right next to the ICU, right next to the IPD. The challenge is nanopore sequencer has its own problems with errors and everything. So then we said, you know what, let us build the kit so that we can process the sample where we have control over the amplicons that are getting done. We have a much higher depth of sequencing. We have significantly higher fidelity on the polymerases that are being using on the entire process. So we made a kit. The nanopore sequencer is already available. It's an inexpensive device. And we have that entire software in which once the sample is processed, you land it on the nanopore. And today, I'm really happy to say more than 15 hospitals in India who, till we reached them, they were only doing RT-PCR. They are today running nanopore sequencing for detecting sepsis right next to their ICU, direct from blood. The best blood test for RT-PCR that is available in the world today from Biomiru, even that has a culture step, and then they go ahead and sequence on BCID. This is direct from blood on a nanopore. Result starts coming out in the first 6 to 8 hours. It is unheard of. And that is the difference. The bioinformatics that we created is common. The genesis of the problem was tuberculosis. We started detecting different bacteria, but the deployment is wildly different. Tuberculosis is working on short read sequencing in a hub and spoke model to get benefits of scale. Sepsis is working at edge, on edge genomics, in premises of hospitals to get benefits of turnaround time.
Wow. Wonderful. I see a lot of activity during this. I will ask Shruti, do you want to ask your own question or should I read it out to you?
Please go ahead.
Okay, so it's a question from Shruti. You can turn on your camera if you are in a position. So what Shruti is asking is, please provide your comments on introducing NGS intervention towards mitigating the use of antibiotics as drugs. Should this tech be used for tracing AMR gene among human populations across India?
Yeah, Shruti, that's a very good question. And it is something which is close to my heart. And yes, I mean, see, the gut microbes are a potential source of antibiotic resistance, as well as it's a environment where a lot of horizontal gene transfer takes place. So there has been a lot of interest in looking at the AMR pattern in the gut, and I cannot disclose the results right now, but the pan-India gut microbiome project that we have, we are also looking at AMR pattern of the individual volunteers in this project, and which spreads across 4 biogeographic regions of the country and 17 communities, including some around 3,500 individuals. So once we, and these are from different socioeconomic strata, villages, rural, semi-urban areas. So once we have that data, I would be in a better position to comment on what is the AMR pattern at different populations in India, whether the tribals, which we presume that they would not have come in contact with any antibiotic exposure in their life, whether they also still have any antibiotic resistance genes in their gut microbe. I will be in a better position to make comment on once we have the metagenomic data on those patients, which we have recently received. So we are analyzing that data. But it's a very good question, and it's a very important point to consider.
Thank you, sir. Sir, I had something extended to my question just now. I was wondering to ask you, if
Sure.
that's okay to ask you,
Yeah, please.
that will it be, do you think that it will be necessary after your research comes out? Will this be necessary at certain point that all the medication that doctors are giving, that like the antibiotics, the AMR gene data before prescribing any antibiotics to the patients? I'm just wondering if such research comes out, the way the doctors are delivering drugs to the patients will change in clinical respect. Thank you.
See, once the results of the research are out, there are many ways this information can be used. One way is also, for example, I mean, this is a imagination, but one way is supposing you find that a particular individual gut microbiome has a very large number of antibiotic resistant genes, then it may also be possible to develop some fecal microbiota transplantation or use of some consortia to replace this not so good microbiome, which may be otherwise okay, but contains a lot of antimicrobial resistant genes. So it may be possible to replace this microbiome. Another possibility is that if the AMR is developing, I mean, these are all futuristic things. If the AMR is developing during the course of your life, so more and more antibiotic exposure is leading to more and more antibiotic resistance in your gut. There is a concept of fecal microbiota bank. So you store your own fecal microbiota at the earliest stage of your life, and subsequently you develop such problem of having AMR diarrhea or in general AMR microbiota, then use your own fecal microbiota to replace replace your older and healthier microbiota. So there are many such possibilities as we go further in this research and more technologies develop.
Thank you, sir.
Next question is by Professor V.S. Chauhan. He's asking, despite the understandable push for technological advancements and the potential for faster surveillance of AMR using technologies like Haystacks has, it remains a challenge that no pharma industry, at least in India, is showing an interest in developing new antibiotics. So in this light, how should academics proceed with promising leads?
Can I answer, or Anirvan is going to answer?
Both of you.
So my answer to this is, on one hand, it is not just Indian academic industry, it is a pharma industry, it's global. Because what has happened is there are two reasons for this. So I was told, one reason is that you know very well that the investment for anything for the human consumption.
We know the reasons. Can I just say that we know the reasons? These are published, they are printed, they're advertised. So I don't think that is the answer I'm looking for. I'm simply asking, technological advancement, giving the future, we'll do this, such huge variables in in the public. We know exactly what happens in developing country. It doesn't have to be in my gut. It is in the population. AMR can develop in population. There is no escape. So I pointedly asked this question because the funding agencies are funding molecules to be developed as a new antibiotics and so on. If all of us agree here that the pharma is simply not interested, so are we just beating around the bush by doing sequencing? By doing understanding, fine. You want to publish a paper, how AMR develops, nobody minds that. We know AMR develops for the past 70 years. So I think we are skirting around the problem of how do you stop people from dying when they have antimicrobial resistant bacteria in their body, in their lungs.
But I don't think the solution is to develop more antibiotics.
How do you do that?
The world is going to develop more antibiotics in many different ways. There are fast therapies and several other things that people are working on. The question is, sir,
Adjuvants for existing there is also
yeah, exactly. So I think
adjuvants for existing antibiotics.
a lot of this work is going on. The big question is antimicrobial stewardship. How many times? Because see, at the end of the day, what we need to understand is, and I don't think we are talking about doing genome sequencing for publishing data. The idea is when we do genome sequencing, we understand the scope of the problem. To connect this to the previous thing, like when the question is asked, how does a clinician give antibiotics? A clinician doesn't even wait for a microbiological investigation to be over to give the antibiotic. Antimicrobial stewardship, if we don't do this, it doesn't matter how many antibiotics that we develop, it's only going to develop more resistance. We already have bedaquiline resistance in the population. There is colistin resistance in the population. I understand your point that there has to be more effort in developing more antibiotics. But I think till such time that we find a platform, so what we are talking about in platform technologies, can we have a platform where large number of molecules and say biomolecules, biotherapies can be tested and validated to take forward? Maybe somebody is working on that platform. But till such time, I think antimicrobial stewardship programs, which are right now limited to hospitals, need to be expanded to countries. Because AMSP is not the responsibility of the Apollo Hospital or the Max Hospital. AMSP is the responsibility of the entire healthcare ecosystem that we are talking about. So I personally don't think developing more antibiotics is a solution.
I don't want to debate this. I just want to make a final comment. I think, I don't know where everybody lives, if we all live in India or live in Bombay or Delhi. I think if our feet are firmly on the ground, then I think we will have a slightly different discussion. I just go across to my chemist here and can get any antibiotic that you and I might develop. So I agree with you. Developing, it's not a question of whether developing new antibiotics is a solution or not. The question is, you can push technology, you can understand the problem, you can do the surveillance much better than you did before, but you still have antibiotics available across the table. No doctor, how many people end up going to Apollo or Max or Fortis in your view in India? Maybe 2%, maybe 3%. So I think we shorten the problem by not understanding the gravity of the situation. If you don't understand the gravity of the situation, you can understand as much as you want. We've done it for cancer. We've done it for malaria. We have sequenced genome of the parasite. We have sequenced genome of the human. We have sequenced genome of whatever you want. We have sequences, single cell sequencing. But I don't think solution followed from this. I'm not arguing for not pushing the technology or not dwelling. I'm not arguing for that. I think technology is fantastic, breathtaking. But I think our feet should be firmly ground when we talk about AMR and finding solutions for it. My comment, I don't want to debate with anybody, but I think we should not lessen the problem that exists in developing countries.
Thank you, Professor Chauhan, for that comment. Thank you. Masood, you have your question ready. Can you ask it?
Good evening, sir. Thank you for giving me the opportunity to ask the question. My question was to the Yogesh sir and Anirvan sir. Like the rise in the AMR is the major concern for overall all the communities overall the globe, and WHO has taken it as a major concern till 2050. The only thing I wanted to know is the how the antimicrobial peptides will work tackling AMR, as there was somebody was telling that the cost effectiveness of the antibiotics is also rising because of the pharmaceutical industries are not ready to introduce new antibiotics into the market. So how can we utilize this antimicrobial peptide in order to tackle AMR, especially like countries in India where we live? Introducing AMPs are going to be very, not much cost effective. So how can AMP can be utilized if we produce them in a mass? Thank you.
Anirvan, you want to answer this?
No, I don't know anything about this.
Okay, see, Masood, there are many ways in which AMR problem is being tackled, and I made a remark sometime back about use of adjuvants. So AMPs is also one of the therapies that is being thought of, being used, but presently the AMPs have success. From whatever I understand, the AMPs have success only in topical applications, and system infections, so far AMPs have not been very successful. But I presume with the time we will be able to develop more targeted approach for delivering AMPs in the right locations, and the technologies will improve. So fast therapy also, again, there was a mention of fast therapy. So fast therapy also today is being used in a limited way because of the limitation of strain specificity of the phages. But I hope that with the time these technologies will evolve and they will become more specific and more effective.
Mansi, you can go ahead next.
Thank you, sir. Thank you.
Yeah. So I have a question to both Dr. Shouche as well as Dr. Chatterjee. I was just wondering that I agree that NGS is a very good solution to understand what is happening with AMR, but sir, it is not very cost effective, right? And for diagnosis of a prompt decision making of what antibiotic to use and what antibiotic to exclude, we will need to, I believe that we would need to have a diagnostic or a prompt diagnostic solution like qPCR based assays to identify the escape pathogens and AMR or ARGs, right? So, but when you come with this idea, it is very difficult to convince the clinicians and the microbiologists who are working in the hospitals. They usually say that we go by phenotypic assays. We look at how the organism is growing. Then they'll ask you a question like, what is the difference between a commensal and an infectious agent? They could see that and then use their judgment, and it comes to the thing that, oh, we have experience and we know this, and there is a lack of there is kind of a inhibition to adopt to molecular surveillance using qPCR. I want your advice and your comments about how do you look at this, so yeah.
Sir, may I take this question?
Sure, please.
Professor?
Yeah.
So it's very close. This is practically my bread and butter right now, because everybody asks the same question. Mansi, just a quick question back to you. How much do you think an NGS-based diagnosis of a pathogen in a hospital would cost?
So what I have calculated, I might be wrong, but I think it should go until like 4,000 to 5,000 rupees, if I'm not wrong. No, it's cheaper than that. I am not sure, sir. Because I am trying to develop this qPCR based assays, and hence the question.
So now, when you talk about a 4,000 to 5,000 rupee assay on your own terms, I was told this by Professor Sanjay Bhattacharya, and this was about 1.5 years back. He's the head of microbiology at Tata Memorial Center, Kolkata, and he had asked me this question: who is my competition? And I had thought of many different things. This is my competition, that company, this. And he told me this very enlightening thing. My competition is the hospital bed. That ICU bed costs the patient several times more than a test like this. Now, when you connect this problem of ARGs and MICs, absolutely. The place where bacteriology is and the way microbiologists are using it, I think it is very difficult to change behavior. But the fact of the matter, Mansi, is, again, this is something I learned from a doctor. This is Dr. I think Todi in AMRI Kolkata, who once said to a microbiologist, and I was sitting there, that you tell me the patient is dying on the bed. I, as an intensivist, knows that the patient has sepsis, and culture 90 out of 100 times tells me there is no infection.
Yeah.
That is the sensitivity of culture. So what we are trying to understand is why will clinicians, to answer your question, a clinician actually does not care less about whether it is microbiology, whether it is genome sequencing, whether it is RT-PCR, or it is some proteome or some AI. A clinician doesn't care. A clinician wants to know what drug to give. The way you tell it is your headache, right? Now, when you look at technologies, you need to look at technologies in two forms. One is screening, and the other is diagnostics, right? When you are trying to dig deeper into it. And this is exactly what we did. This was the theory when we started working on TB in UK. In UK, for TB patients, they would do all the tests, TANS polygotyping and Miru VNTR and all of that. They realized that every time they hit a wall with a certain sample, they would send that sample for genome sequencing. So anything that was not getting resolved by 20 different methods in the labs of UK, they would send it for genome sequencing. So we asked a very simple question: why don't we turn the question on its head? What if we do genome sequencing first? And for everything else where we require deeper answers, we can do the microbiology. And this is exactly how NHS moved into TB genome sequencing. They realized that if they scale genome sequencing, it actually costs lesser than microbiology. So today, the genome sequencing test of tuberculosis that patients access for about 7,000 rupees, it is 50% the cost of 15,000 rupees that 11 drug midget test takes in the real lab. Genome sequencing is cheaper than current day technology. And the moment you talk about RT-PCR, how many targets are you going to put in an RT-PCR? How many different bacteria? So when you work with an RT-PCR, you are entrusting the clinician with the one question which the clinician doesn't want to take a decision on. The clinician is entrusted with the decision which test to perform. Am I looking for this? Am I looking for that? Am I looking for that? That becomes the entire paradigm of diagnosis. कौन सा test order किया will determine how the patient is going to respond. Whereas if you do genome sequencing, you don't need to decide which bacteria or fungi to look for. You can actually do genome sequencing, identify species, strain, sometimes even substrain level information, based on which you can actually take a decision. And if you do it for 100% of the patients, and this is where we are challenging the thought. What we are challenging the thought is, instead of chasing the bug, the RT-PCRs of the world today, the microbiology of the world today is chasing the bug. You first have an infection, you have the fever, then you go and palpate the patient, then you decide that this is a hospital-acquired infection or a community-acquired infection. It is in the lung or in the stomach. Based on all of that, you know, I want to do typhoid, I want to do pneumonia, I want to do this, I want to do that, right? Instead of that, what we are saying is, every morning for an IPD, you go ahead and do a genome sequencing test. You screen for a pathogen. You don't wait for the pathogen to come. When you do this, you imagine like a CBC, if 40 people in a hospital are getting their pathogen sequenced every morning, the cost of genome sequencing is going to be lesser than RT-PCR. And that is the change we want to see, and that is the change clinicians want to see, that is the change the microbiologists want to see, because today the microbiologist, despite doing all the tests in the world, is not creating an intervention for the clinician who is so deeply dependent on that microbiologist. When you have the right solution, it is cheaper, it is less expensive, and it is better. Yes, sir, Professor.
No. I just wanted to add to what you have said, giving a very simple example. It's a question of demand. You know, as the demand in the market increases, the cost will come down. And the best example, again, probably this generation, younger generation won't know. But when the mobile came into the market first, the set was costing 20,000 rupees, and per call per minute cost was something like 125, 150 rupees. And you see that today, just because of the sheer demand in the market, how much the cost has come down. Now almost everybody can have and afford a mobile. So same way, these technologies, as the demands increases, the technology will improve and the cost will come out dramatically.
Thank you. Thank you.
Can I
the whole point is it has already come down.
Yeah.
And this
it will go down further. My point is, it will go down further.
And this is what I meant by saying that we are sitting in the midst of godfather of metagenomics and genomics. Because of many of this work that has happened, the application has increased. Because applications has increased, use has increased. Because I did the first TB genome sequencing in 2011 for 1 lakh rupees, one sample. Today, a patient is accessing that test for less than 7,000 rupees with all the built in. That's the change.
Yeah, I will move on to the next question. It's by Suraj Shukla. He asks, which technique is more reliable among 16S rRNA, multi-locus sequencing typing, that is MLST, and ribosomal MLSTs for bacterial identification at the genus or species level? Can kindly anyone give inputs regarding identification?
Yeah, I'll answer this quickly, Suraj. See, it depends on the organism in question. Today, because of the limitation of 16S rRNA gene sequencing going to species level, depending on the genus, there are many additional MLST schemes which are genus specific. So if 16S rRNA is inadequate for your genus to go up to the species level, then you use appropriate MLST schemes. But with, as you know, that with genome sequencing technologies becoming cheaper and cheaper, even MLST would be obsolete, and one can straight away go for genome sequencing, which will give you much more accurate information up to the strain level, not just species, but the strain level.
Thank you, sir.
Rahul, then followed by the Surojit.
Yeah, hi, this is Rahul Roy from IISc. I had a few questions specifically for Anirvan because he has a lot of experience doing this for diagnostics in a commercial setting. So Anirvan, I wanted to understand, you talked about some price points. I was curious, what you think is the price point where you think there will be effective adaptation from the market? Second is, have you started seeing any competition from mass spec proteomics, which is being touted as a 200 rupee test for detecting AMR and bacterial infections? And the third question was about sensitivity. You mentioned sepsis you are able to detect. I wonder what is the CFU per ml that you are detecting? At what point are you starting to detect using nanopore? And whether you can detect earlier, even before the situation worsens to a state where you have clear signs of sepsis. Thanks.
I think tough questions there. Cost. As Professor Chauhan was saying, right, there is a Max Hospital, and then there is rest of India. I think so our theory is, and this is what we are implementing. Our theory is that adaptation will take time. It's a chicken and egg. The more we adopt, as Professor Shouche said, the more we adopt, the cheaper it will get. So we start with critical care. I think in critical care, the price point where we are already at a range between sub 10,000 rupee range in critical care, in ICU patients, this is already acceptable in the market. It will get about 30 to 40% cheaper in the next 2 years. This will get 40 to 60% cheaper in critical care. The moment it gets 40, 60% cheaper in critical care, once this comes to that 2,500, 3,000 rupee range, that is where IPD comes in, and the hospital ecosystem is taken care of. At that point, I think the price point in a hospital will be around 1,200 rupees a test. By the time it reaches GP, where you're looking at 1 billion plus tests a year across the world, and actually a few times billions per test a year, sequencing is possibly going to sit at a $20, $15 to $20 kind of range as a price point. But the market is not going to adopt it in one go. It is going to adopt it first for critical care, then for a slightly wider usage, and then in the GP network that it is going to be doing. And I think it will settle down to that kind of a pricing. The pricing will also depend on the market because some markets are factored in by the insurance and others are out of pocket. But this is where I think the cost will settle in part. Absolutely, there is competition from mass spec. The most interesting part about mass spec and some of this technology that the cost of the technology is not even in rupees; it is actually in paise. It is something which is very much there in the market. We don't particularly see a lot of headwinds on it because again, the problem has become so much more complex with antimicrobial resistance, with antifungal resistance, that some of these outcomes are yet difficult to be caught up with a mass spec and a MALDI-TOF kind of a solution. Having said that, they are there. We have competition. I think we will be able to settle down on that as well, but it will take about 3 to 5 years to figure out how this will settle. Because I think the way the market will move will be in two parts. One is a POCT, where we think that, and this is not wishful thinking, this is exactly where we are working towards. We think that for a given location, for a given season, there will be home testing kits where the flavor of the season, right? The pathogen of the season, the ARG of the season will be available on a strip and people will be able to test it at home. That will take care of 90 to 95% of infectious testing that the world will be doing. The 5% who end up in the hospital will definitely go in for genome sequencing. This is my best guess. Please don't take me up on it, how the market moves. But having stayed in the market, this is how I am seeing. Sensitivity, fantastic question. It's not a one size fits all. There are some organisms which we are able to pick up at less than 2 CFU per ml. Strep, staph, suits, we are able to pick up at very, very low CFU. But there are others like acinatos and some other fungi that we are seeing that we are able to take around 10 to 12 CFU per ml. That is where we are seeing it is still an evolving thought on our evolving data on our table. We have covered now about just less than 100 different kinds of organisms, having been in the field, and we encounter a new one every day, and then we put it on the validation scheme. But yes, it depends on the time of collection. It depends on the exposure of antibiotics that the patient has had. And it is the kind of bacteria or fungi that we are looking at. In general, a very mean number that I want to talk about is 10 CFU per ml. We want to hit that consistently, primarily because the amount of blood that we are drawing already is becoming detrimental to move into neonatal sepsis. So in neonatal sepsis, anything more than 500 microliters of blood is very difficult to have. And currently we are working with around 4 to 5 ml of blood. With 4 to 5 ml of blood, with 1 CFU per ml, 10 CFU per ml, there's a lot of bacteria to talk about in terms of genomics. But yeah, that's broadly the answer that I hope I was able to touch on most of the points that you asked.
Yeah, thank you so much. Yeah, very informative.
Thank you, Professor. After that, Surojit.
Good evening, very much good evening. And this is a nice discussion going on the sequencing platform and the antimicrobial resistance. My question is that for analyzing the antibiotic resistance genes and also the plasmid incompatibility types or INC types and the virulence gene and also the whole genome sequence type, that is the MLST, whole genome MLST, plasmid MLST, for doing all these things, which sequencing platform should I use and how much data should be generated, and also suggest for the pricing.
Anirvan, you can handle this.
So on the platform, sir, would love to hear from you on because it's much wider than what I work on. I can definitely come on the pricing, but on the platforms, your thoughts, sir, between whole genome and
if you want
virulence.
to capture, Surajit, if you want to capture all the plasmids and all, you really need to go for deep sequencing. And probably I would suggest that if you want to do a in-depth sequence analysis, including mapping of all the plasmids and the genes on the plasmids, the hybrid approach is the better approach, where you use a technology like PacBio or nanopore to generate longer reads, so that you can generate the scaffolds on which you can use high resolution data like Illumina to map your high resolution data on those scaffolds. So if you want to go that deep and that higher resolution, hybrid technology would be a better. And of course, that high resolution comes with a cost. So it is going to be expensive. When you use a hybrid platform, both the technologies, very high resolution sequencing, it is going to be costly. I would put the cost in the tune of, if you want to do a real high resolution sequencing, it will be above 50, going up to probably 1 lakh also for a single genome.
For a single genome.
Yeah. Am I right, Anirvan?
Absolutely. Absolutely, sir. It's amazing. I had almost forgotten about hybrid sequencing, but yes, that's absolutely what you would want to do. Amazing. I think also, Shurajit, costs are subject to time. So the faster you want the result, the more expensive it is. If you can wait, some of these costs can actually come down. But my suggestion, in addition to that, is when you're looking at cost, don't look at cost of one genome. You need to design your experiments well to know what you are sequencing for what. Once you have a lot of genomic data, it just becomes a huge haystack, right? And to find that one needle, it again, as was said, you will get a paper, but you may not be able to answer the question that you were asking.
Actually, my target is to identify the resistance gene, as well as the virulence gene, as well as the sequence type or ST type. And for that purpose, 2 GB data is sufficient if I using
No. 2 GB data is nowhere to where you would want. Yeah, sorry. I can already see Professor Shouche is also nodding his head.
Yeah. No, 2 GB is very less.
Okay.
I will sort of combine two questions which have been in the chat. So one is, I guess this is for haystacks particularly, and Dr. Anirvan, that what would the report contain? Just an organism or AMR list of AMR genes? And the second is, can we really do away with cultures if we need information on AST?
No. So the answer to that second question is absolutely no, for the very simple reason that MICs are very important in clinical decision. And that's why I wanted to make the difference with, and I have a very nice, very real story last week. A patient who was part of a study in a hospital. The patient was tested. Okay, so the patient was tested on standard of care, which is a culture, and about 24 hours later, we collected the sample for the sepsis test on sequencing. Now, we generally say the sample must be collected in parallel to be able to compare it better. But this was after 24 hours, and I was shocked that we had actually detected an organism with ARG. So we went back and asked, what happened? And very interesting case here. The culture had flagged positive and antimicrobial resistance with MIC of 8 was detected. Don't remember the drug, colistin or something. And the doctor had started the therapy accordingly. Now, it so happened that MIC was not very effective. But when it came to sequencing, we just found ARG. We don't care much about MIC. And we said it's not going to work. So basically, we were able to pick up the bug because the antibiotic was not very effective. The bug continued to be in circulation, and we were able to pick it up. So it's both sides. I want to make it very clear, at least for the next 2 years, I don't see RT-PCR or molecular methods in sepsis management to replace culture, because one of the most important pieces of information a clinician has needs, especially in current day, is MIC. What concentration? So it will not go away. It will be there. What will come in is going to be a better screening test, which is faster and wider. So I forgot the question, but I think this was the question.
Yeah, the other component to that question was what would the report contain? Just the list
Oh.
of organisms.
No, so
organisms.
This will be the report contains. So genomic reports are very interesting. They contain a lot of audit information. So now what it generally has, first line is pathogen identified. If there are multi-pathogens, say a bacteria with a fungi, which is not uncommon, both of them will be there. Then there is the depth of sequencing, the coverage of sequencing, the mutations that are found, the gene, all of that comes on the second page. On the first page itself is organism and nothing else. Then comes the entire sequencing details, or the ARG, the class of ARG, the drugs associated with it, and generally with sequencing, again, what is there are more audit information. What was the flow cell? When was it run? So all of these information is there. The version of the software that was used, the version of the catalog that was used. So it's a very dynamic thing. It is completely automated, just like any other software you would have. It's completely automated. But headlines are, yes, pathogen/S, pathogens, ARG/ARGs, coverage, depth, version of software, version of catalog, and this rest of the audit details.
Mansi, can you quickly ask your question?
Yes. So, sir, reiterating on a previous question that I had asked, sir, what is your views on the biofire panel which exists in the hospitals? The reason I'm not anti-sequencing, I do myself sequencing in the lab, but when I try to go and talk to the clinician, they are like, we are scared with the amount of information it gives us. Even if there is no pathogen in the blood or in urine, we will get to this pathogen, we'll start an antibiotic to it. And so they're very reluctant to use the biofire glow panel. The reluctancy is two ways, sir. Firstly, there is a lack of, not all hospitals can use it because not all hospitals are very well equipped that way. The second thing is, it's very expensive. So per patient cost is like 18,000 rupees. But again, we spoke about it that it is basically an ICU comparison. So the market is different, right? But how do we convince because a genomic report gives a lot of information, right? Lot of pathogens, lot of fungus, lot of bacteria. How do you think can we convince the clinicians to adopt? Okay.
So Mansi, you're absolutely right. It can be overwhelming, but that is precisely where good data analytics come in. So when you use good data analytics, see in a blood sample, and I was enlightened on this by my colleague, Dr. Mahua, who is an MD microbiology from PGI. She said her professor had once said that God is not so cruel that somebody is going to have 5 different infections at the same time. I mean, it's not the best way to do science, but unless it is a leakage of the gut, unless it is a burn patient, blood is unlikely to have multiple pathogens. It is not that it doesn't have, there are. So, but then that is where analytics comes in, that is where sequencing comes in. It can be overwhelming, but I think if the technology is done right, it is not. So we haven't faced that problem in what we are working on. But on BioFire, if you have used that machine, while they are my competition, I must tell, it is one of the most beautifully engineered solution I have seen. The way they have integrated microfluidics with the film array and all of that, it's a beautiful technology. It's a fantastic technology. It's very expensive, and we will end up replacing it. That is pretty much it. The clinicians don't use it primarily because it is still after culture. BioFire is working very well for respiratory panels. The clinicians are using BioFire for respiratory panels because those panels are direct from sample and the reports come very fast. In case of blood, the BCID2, it is still after culture. And that is the primary reason why BioFire is not used, apart from cost and all of that. And this I was told by one of our mentors at Sign. When you are in healthcare, cost is a huge factor, but the bigger factor is value. Can you provide value? Right now, the BCID2 does not provide value for most of the clinicians and most of the patients. And that is the primary reason the adoption is low.
Thank you. We are sort of over time, but I have one question, last question from like an in-house question. If anybody can answer, the question is, if people want to use sequencing as the core of the technologies and come up with a commercial product, what sort of help would be required from, let's say, people from ecosystem enablers like CCAM? What is that small, or what is missing in this current scenario that would enable the use of this very beautiful technology, as you mentioned, to come up with more defined products?
Sorry, I'll go first. And I think, Ruturaj, as an entrepreneur, whenever I hear problems, I look at opportunities. So it would be great to have a lot of help. And if you ask an entrepreneur, we will go on a laundry list of things that starting from money to hospital connect, to scientist connects and all that. But frankly, the issue is not sequencing. The issue is not all of that. If we can together drive the appreciation of the intensity of the problem, because what we are losing is time. And for that, we need help. You know, what Professor Yogesh and his team and other scientists are doing in the world today, that's all fine. What I'm trying to do is also fine. But we are not respecting time. From the time I started TB solutions, TB genome sequencing, when my first paper with Haystack, I did not have any bedaquiline resistance. The latest part, I'm already seeing resistance, and the world is seeing resistance. We are losing time. Can we expedite the transfer of knowledge and get the clinicians and others to work together? And it is not a difficult thing. If you say that these are the 10 hospitals where all the current technologies will come and start working. If we do that, I tell you, the biggest adopters that I have come across in the healthcare system are clinicians. They are crying out loud for solutions. Professor Yogesh and others, they have solutions. Where is the bridge? That is what we want, just to reduce time, nothing else.
Thank you. We are slightly over time, but I couldn't stop the session considering the passionate involvement of everyone. I thank you, Professor Yogesh Shouche, for agreeing to be part of this monthly webinar series, as well as Dr. Anirvan Chatterjee. Thank you very much for your inputs. And I thank you, the participants. We had over 100 participants. I thank each and every one of them in making this webinar a success. Thank you.
Thank you. It was a pleasure to be here. Thanks a lot.
And it was my honor to be here with you, sir, again. Thank you.